Hello, I'm Rabab, your audio navigator, and here I will guide you about immunology, that is the sixth section of the biochemistry syllabus. It constitutes a very important part of the syllabus, and many questions are asked from this area. Questions asked are generally concept based, so you need to go through this in detail, covering all the important facts and basics. Questions asked could be of 1 or 2 marks. So I'll start with immunity, both active and passive. First of all, you should know what is immunity. It is nothing but the adequate biological defense mechanism to fight infection diseases, and also should have adequate tolerance to avoid allergen autoimmune diseases. So should be well aware of all these terms. Firstly, the immunity could be divided into two groups, that is adaptive immunity and innate immunity. So you should be well aware of both these kinds with examples and how are these attained. Then after that, the adaptive immunity could be natural or artificial. So you need to cover these two also with examples. And the adaptive immunity could also be divided into passive and active, both natural and artificial means. For example, natural immunity could be passive if it transferred from maternal side, or it could be active if it is acquired by some infection. Then the other one is the artificial one. It could be passive by antibody transfer, or it could be active by the means of immunization. So you need to cover all these kinds with in detail with many examples. And you also need to cover the historical background of the theories given by different scientists regarding immunity. What were the different breakthroughs in this field? Who were the main scientists in this field, and what were their contributions? And the development of different vaccines. The vaccines could be traditionally divided into four types. For example, the inactivated vaccines, then there are live attenuated vaccines, then the toxoids, and then the subunit vaccines. So you need to go through all these with examples. After this, you also need to study about the complement system. What is the complement system and how is the complement system activated, and what happens as a result of the activation. So these are the topics which you need to cover in this. And also you should know about the historical background. Then what are the immune functions which are triggered by the complement? For example, the phagocytosis, inflammation, and membrane attack. Then what are the three different pathways by which the complement system may occur? For example, the classical complement pathway, which is triggered by the activation of the C1 complex. So you should know what is the C1 complex composed of. Then what are the different steps involved in this pathway? Then what is the alternative complement pathway, which is continuously activated at a low level as a result of spontaneous C3 hydrolysis due to the breakdown of the internal thioester bond. So we need to cover this pathway in detail with all the steps and what are the different factors and enzymes involved in this pathway. Then what is the lectin pathway? What are the factors which are involved in this? Then how does the activation of complements happen by antigen associated antibodies? Then how does this complement system is regulated? Then you also should know about the diseases which are associated with the complement deficiency. For example, the Barker-Simmons syndrome, asthma, lupus erythematosus, then glomerulonephritis, various forms of arthritis, autoimmune heart disease, multiple sclerosis, inflammatory bowel disease, paroxysmal nocturnal hemoglobinuria, etc. It could also lead to Alzheimer's disease. So we need to cover all these. Then what are the effects of deficiencies in complement regulators? So also cover this briefly. Then comes the antibodies, which are the proteins synthesized by an animal in response to the presence of a foreign substance. They are secreted by plasma cells, which are derived from B lymphocytes. These are responsible for the humoral immune response. So you should know that the specific antibody is synthesized following the exposure to an antigen. Then how are these antibodies formed? They are formed by selection rather than by instruction. So you need to cover this whole mechanism. Then what is the structure of antibody? It consists of distinct antigen binding and effector units. What are these Fab and Fc region? Then how are monoclonal antibodies prepared? Then what are the light and heavy chains of antibodies? And what do they consist of? Variable and constant regions. What are the homologous domains of immunoglobulins? And what are the genes which encode the variable and constant regions of the antibodies? They are encoded by separate genes and then they become joined. How the variable regions of L and H chains are encoded by several hundred genes. How the antibody diversity is increased by the J and D genes. So you should know all about these and what are the facts related to this. How different genes encode and get joined to form the antibodies. So you should know vaguely that how much antibodies could be formed by the combinatorial association and somatic mutation. They are around 10 to the power 8. Then what are the different kinds of antibodies? Different classes like IgG, IgA, IgM, IgD, IgE. Then you should know what are the mass of all these. Then their chain structures and the light and heavy chains which they consist of. Then what are the different functions which they perform. Then how the different classes of antibodies are formed by the hopping of VH genes. Then after that, what are the different cells which are involved in the immune system, like the T cells, B cells and the macrophages. So what are the killer T cells? Which destroy the infected cells that display foreign peptides bound to class 1 MHC proteins. So you should know what are these terms referring to. What is the pathway by which the class 1 MHC proteins on the surface of the cell display peptides that are formed by the proteolysis of cytosolic proteins? Then what are the helper T cells and how do they stimulate B cells that display foreign peptides bound to class 2 MHC proteins? This is a bit complicated, so you need to clear your basics. Go through these pathways in detail. You should also know what are these major histocompatibility complex MHC proteins that are highly diverse presenters of antigens. What are their structures? What are T cell receptors and what are their structures? They are the antibody-like proteins which contain variable and constant regions. You should also know what are the genes which are involved in their expression. Then what are CD8 and CD4 molecules? They are found on the killer T cells and helper T cell respectively. Then what are their functions? They act in concert with the T cell receptors. So you need to go through the whole mechanism. How do they function? I should also know that what happens when the human immunodeficiency virus subvert the immune system by destroying the helper T cells. So you should know what are the effects which it causes. Then after T cells, what are B cells? What are the B lymphocytes? Then how do they create plasma cells and B memory cells? What is the mechanism by which they are activated? Then what is the role of helper T cells in eliciting B cell response? Then after that, what are phagocytes? And what are the different types of phagocytes like granulocyte, macrophages, and dendritic cells? What are the roles of all these, especially the macrophages? How do the monocytes turn into macrophages? So for this part, you need to know how are T cells, B cells are developed. Then how do the activation take place? Both, like for B cells, it could be the T cell dependent activation or the T cell independent activation. And how does the memory B cell activation takes place? So you need to know all the mechanisms in detail. What are the molecules and enzymes involved in it? And for the activation of T cell, what is the role of CD4 plus T cells? What are the role of helper T cells? Then what are the different interferons and IL molecules related in this pathway? Then what is the role of cytotoxic T cells or the CD8 plus T cells? So you have to study all this in detail. Then what are major histocompatibility complex? They are the cell surface proteins essential for the acquired immune system to recognize the foreign molecules. You should know about its structure. And what are their functions, like antigen presentation, autoimmune reaction, tissue allo recognition, etc. Then what are the different classes of MHC molecules? They could be of class 1, class 2, and class 3. Mainly the main important groups are the class 1 and class 2. Then what are their roles and what are the genes which are involved in the expression of these proteins? Then what are the two antigen processing pathways which they exhibit? The MHC1 pathway and the MHC2 pathway. What are the different steps involved in these? You also need to do the comparative study of both these pathways. Then what does the term MHC restriction refer to? What role do they play in the transplant rejection? In humans, they are also known as the HLA. So these rejection could be hyperacute rejection, acute cellular rejection, or the acute humoral rejection and chronic dysfunction. So go through all three of these with examples. Also need to go through the HLA biology. What are the different genes which are needed for its expression? What is MHC halotype? After this, you need to go through what are T cell receptors. What is the structure of a T cell receptor? Then how does the generation of TCR diversity occur? And what is the TCR complex and TCR co-receptors? Need to go through all these terms. And what is its role in the T cell activation? What are the signals which are implicated after the TCR triggering? So you need to cover all these pathways in detail. And that was all about the immune system. Then you need to cover a few immunological techniques. Immunological techniques utilize mainly IgG, which constitutes 80% of the serum immunoglobulin. Then you need to know about the precipitin reaction. What are the preparative uses of the antigen-antibody interaction? Then there are few protocols which are usually followed for immunological techniques. For example, the production of antibodies, and for that production of antisera, which leads to the production of polyclonal antibodies. So what is the procedure for that? Then what is the procedure for the production of the monoclonal antibodies? Then what is the principle on which the precipitin reaction in freeze solution is based upon? Then what do the zones depict, like the zone of antibody excess, zone of equivalence, zone of antigen excess? Then how do the qualitative and quantitative analysis of antigens are done? What is hapten inhibition test? These were the few important topics which you need to cover before moving towards the main techniques, like the immunodiffusion. What is the principle? And well, it could be single or simple immunodiffusion, or it could be double immunodiffusion. You need to know the principle of both of these, and what is the procedure. Then what is immunoelectrophoresis? Well, it could be qualitative immunoelectrophoresis or the quantitative one. Then there is one crossover immunoelectrophoresis also. So you should go all of these, and you should know the principle and the procedure, how it works. And what is two-dimensional electrophoresis, how it is performed. And then how does the visualization and recording of precipitin lines in gels are conducted. Then after this, what is radioimmunoassay, the principle on which it works. Then what are the practical aspects related to it? What are the methods of labeling? Then what are the major advantages and disadvantages of this technique? Go through them. Then the next technique, which is the enzyme-linked immunosorbent assay, that is ELISA, which is a very important technique of biochemistry. So first of all, you need to know the principle on which it works. Then what is the procedure and protocol which is followed. Then what are the practical aspects related to it. Then what is dot immunobinding assay, you need to go through this also. Then finally, what are the applications of ELISA. So go through this in detail. Then what is fluorescence immunoassay, what is the principle behind it, and what are the different kinds of FIA. For example, the homogeneous substrate labeled fluorescent immunoassay. Then there is detailed enhanced lanthanide fluorescence immunoassay. So just briefly go through these two. Then what is flow cytometry cytofluorimetry and fluorescence activated cell sorting, that is FACS. So you should know about the principle on which it works. It is again a very important technique. Then what are bioluminescent immunoassays? Then what are evident biotin mediated immunoassays? Just briefly get the idea of all these. So these were the few important immunological techniques which you need to study, mainly their principles and their applications. So that ends the topic of immunology. Time taken for its preparation could be around 2 to 3 days. Again, I will stress on to clear your concepts regarding these topics. And I hope the information which I shared is going to be helpful for you. And I wish you good luck. Thank you.